Welcome to The Explainer. Today, we're digging into the story of the revolutionary tool that first let us create a DNA fingerprint. We're talking about the microscopic detective work that completely changed the game for medicine and forensics.
Okay, let me ask you this. Imagine I hand you two sets of an encyclopedia. Each set has 3 billion letters, and your job is to find the handful of tiny spelling differences between them. Where would you even start? Well, for decades, that's exactly the problem scientists faced with the human genome. So here's our game plan. First, we'll tackle the problem itself. Then, we'll meet the amazing molecular scissors that provided the solution. We'll see how those scissors create genetic maps and unique barcodes. And finally, we'll look at how this whole thing changed the world.
All right, first up, let's really get a handle on just how big this challenge was, trying to find that tiny genetic needle in a massive DNA haystack.
Get this, 99.9% of your DNA is the exact same as anyone else's on the planet. But that tiny, little 0.1%, that's everything. That's what makes you, well, you. So the real trick was figuring out a way to ignore all the identical stuff and zoom right in on that tiny fraction that makes us all different.
So, how'd they do it? Well, the answer came from a place nobody expected. It came from a tiny bacterium's ancient war against viruses.
And here they are, the heroes of our story, restriction enzymes. You can think of them as tiny, programmable scissors made of protein. See, bacteria evolved them as a defense system. When a virus injected its DNA, these enzymes would find it and just chop it into useless little pieces. And scientists had this incredible light bulb moment. What if we could borrow those scissors?
And what's just wild is how quickly this all happened. It went from a strange observation in the 50s to a full-blown, world-changing technology in just about 20 years. By 1978, the guys who figured it all out, Arbour, Nathans, and Smith, were getting a Nobel Prize for it. That's how big of a deal this was.
So now you've got these incredible molecular scissors. What's the very first thing you do with them? You start making maps. You start charting the massive unknown territory of our DNA.
This whole process is called restriction mapping. Imagine you have a long, blank piece of string. If you use scissors that only cut at red dots, you can figure out where all the red dots are. It's like creating a road map for a highway you've never seen before, just by looking for the mile markers.
Let me show you a really simple example of how this works. You take a long piece of DNA, cut it with one enzyme, and you get a few fragments of specific sizes, okay? Now take that same long piece and cut it with a different enzyme, you get a totally different set of fragments. But the real magic happens when you use both enzymes at once, which gives you a bunch of even smaller pieces.
And here's the clever part. The smaller pieces you got from using both enzymes have to fit perfectly inside the bigger pieces you got from using just one. It's basically a logic puzzle. By seeing how all the little fragments add up, scientists could figure out the exact order and spacing of the cut sites. They could draw a map. Okay, this is where we get to the heart of it. We're about to go from mapping one random piece of DNA to actually comparing the DNA between you and me. This is where the genetic barcode comes into play.
The technique is called RFLP, or RFLP. It stands for Restriction Fragment Length Polymorphism. I know, it's a mouthful, but the idea is actually really simple. If your DNA sequence is even a little bit different from mine, the molecular scissors might cut in different places, and that creates a totally unique pattern of DNA fragments for each of us. It's like a personal barcode written in our genes.
So, how do these different barcodes happen? Well, it usually comes down to two main reasons. On the one hand, a tiny mutation, like a single letter changing in your DNA, can actually break the spot where the scissors are supposed to cut, so no cut. On the other hand, the amount of DNA between two cutting sites can be different. Some people have these long repeating DNA sequences, called VNTRs, that make the fragment a lot longer.
Now you can't just look under a microscope to see this barcode. It's a whole lab process. You get the DNA, chop it up with the enzymes, and then use electricity to pull the fragments through a gel, which separates them by size. Then you transfer that pattern onto a special membrane and use a glowing probe that sticks only to the fragments you care about, and voila, the invisible barcode suddenly becomes visible.
And look, this was so much more than just a cool lab trick. RFLP totally changed the real world. It gave us the power to answer questions about crime and disease that were impossible to answer before.
Let's start with a classic medical case, sickle cell anemia.
We know today that this entire disease is caused by one single typo in our DNA. A single letter A gets swapped out for a T in a crucial gene.
And here's the amazing part. By sheer coincidence, that one tiny typo lands right in the middle of a cutting site for a specific enzyme. So what does that mean? It means the enzyme scissors can cut the DNA of a healthy person, but when it sees the DNA from someone with the sickle cell gene, it can't. The typo literally breaks the scissors.
And this gives us an incredibly powerful diagnostic test. You run the RFLP, and a healthy person will only have the short cut piece of DNA. Someone with the disease will only have the long, uncut piece. And someone who is a carrier, who has one of each, will have both. A simple pattern on a gel could tell you everything you needed to know.
Then, in 1984, came the other huge revolution. A scientist named Alec Jeffreys had a breakthrough. He realized he could use RFLP to look at those repeating sequences we talked about, the VNTRs, to create a pattern so unique to an individual that it was basically a fingerprint. This was the birth of DNA fingerprinting, and it changed criminal justice forever.
Now, let's be real, we don't use RFLP much these days. We have faster, more powerful tools like PCR. But we can't forget where it all started. RFLP was the original decoder ring for the human genome. It was the technique that first taught us how to read the book of life, opening the door for everything that came after.
So RFLP taught us how to read our genetic code, and the technologies that followed, things like CRISPR, are now teaching us how to write it. Which leaves us with a pretty big question, doesn't it? Now that we have this incredible power over the book of life, what stories are we going to choose to write?